Chikungunya, or CHIKV, is a virus that is spread to people by infected Aedes aegypti and Aedes albopictus mosquitoes. The mosquitoes spread the virus by feeding on a person that is already infected with the virus. Once the mosquito has the virus in their system, it undergoes a replication period inside of its midgut to amplify the viral RNA. This then spreads into its secondary tissue, most importantly its salivary glands, and after 2 to 3 days of replication, the mosquito is ready to transmit the virus to a new person through biting. Outbreaks of CHIKV have normally occurred in Africa, Asia, and Europe. However, in 2013, the virus spread to the United States.
A person that contracts chikungunya disease will become symptomatic within 2 to 12 days of being bitten by the infected mosquito. Symptoms include fever, headache, joint pain, mostly in the hands and feet, joint swelling, muscle pain, and rash. These symptoms can occur for 7 to 10 days, and recurring pain can last for months to years, but that is mostly in severe cases which occur in the elderly with pre-existing medical conditions. This can only be contracted once because of adaptive immunity. The antibodies last for a long time. There are no vaccines or treatment for the chikungunya disease, and antigen or antibody tests can be performed to confirm a diagnosis.
When an intracellular virus like chikungunya infects a host cell, hijacking the cellular machinery to replicate and synthesize viral proteins in the cytosol, the MHC1 antigen presentation and processing pathway begins. Viral proteins in the cytosol are degraded into short peptides by a large barrel-shaped protein known as proteasome, also found in the cytosol. These peptide fragments are now able to be trafficked to the endoplasmic reticulum through the transporter associated with antigen processing protein known as TAP. Next is peptide editing, where peptides that are too long to bind to MHC1 molecules in the ER are trimmed of their amino terminus by the enzyme endoplasmic reticulum aminopeptidase. The even smaller peptide fragments are then able to successfully bind to MHC1, and the peptide loading complex and peptide-free MHC1 can be recycled. When a peptide binds to the heavy chain of an MHC1 molecule, MHC1 undergoes a conformational change and dissociates from the chaperone proteins taposin and calreticulin that attach to TAP and the ERP57. This complex, now fully folded, leaves the ER and is exported to the plasma membrane through the Golgi apparatus. At the cell surface, the viral peptide antigen MHC1 complex is presented to cytotoxic T cells and CD8 co-receptors. The CD8 and cytotoxic T cell receptors recognize the viral antigens differ from self and kill the infected cell, preventing further replication of chikungunya.
MHC1 is assembled in the endoplasmic reticulum and is found in all nucleated cells. Thus, the previously mentioned protein proteasome is also found in all normal nucleated cells. The proteasome is necessary in antigen processing because it degrades intracellular viral proteins into smaller peptide fragments. This is important as the heterodimer TAP can only transport peptides, and MHC1 can only bind proteins between 8 to 10 amino acids long. MHC2 is also created in the endoplasmic reticulum, however presents antigens to CD4 helper T cells and does not kill infected cells. Thus, it is important that MHC2 does not bind to the intracellular peptides and components found within the endoplasmic reticulum that MHC1 can target for destruction, and instead only binds to extracellular peptides. This is managed by the invariant chain, which is a protein with the same amino acid sequence found in all humans. The invariant chain blocks the binding site of MHC2 to prevent intracellular peptides from binding and works to stabilize and transport MHC2 as an endocytic vesicle until it may fuse with vesicles filled with extracellular peptides. When the invariant chain is cleaved by proteases into CLIP, then released by the protein HLA-DM, MHC2's antigen binding site is reopened and free to bind and present extracellular peptides to the surface.
Mutations in a person's ability to produce certain proteins can have different effect on how a viral infection is combated by the immune system. A gene mutation in the formation of the invariant chain would severely limit the immune system's response to a chikungunya infection. The invariant chain is responsible for blocking endoplasmic reticulum peptide bonding and moving MHC class II proteins through the Golgi apparatus that then initiate T cell responses to the infection. Therefore, abnormal function of the invariant chain would limit or cease viral antigen presentation. When antigen presentation is limited, it becomes less likely for a macrophage to phagocytize an infected cell. A mutation of the proteasome would affect how a person is able to degrade intracellular proteins. The functionality of this protein is essential to eukaryotes, and the absence of the proteasome gene is lethal. If a person with a proteasome mutation were to be infected by the chikungunya virus, the proteasome might not be able to degrade viral proteins, which are then presented on the cell surface to signal that the cell should be phagocytized. And that is it. Thank you for watching.